Speaking of the Scientific Advisory Board, our next speaker is Dr. Malia Edwards, who actually, not only does she sit on our Scientific Advisory Board, but she's the academic vice chair of our Scientific Advisory Board. She is currently an assistant professor at the Wilmer Eye Institute at the Johns Hopkins University in Maryland. And Dr. Edwards and her lab are investigating how early in the disease process Mueller cell changes occur and how their activation and remodeling affects AMD disease progression by influencing blood vessels, neurons, and retinal pigment epithelial cells. I'm grateful for Dr. Edwards and all the work that she does. Thank you very much.
Thank you, Jess. I just want to kind of reiterate what Kathy said. It's really an honor to come and present at this meeting and have received funding from you, talk to you about some of the work we've done with the funding we've received from you, but just being at this meeting as a researcher is really inspirational and really is a reminder, at least to me, as to why we do what we do. So thank you all, and thank you all for sharing your experiences with us. So I'm going to talk to you today about some work that was funded through the $1 Million Bike Ride Project and the Choroideremia Foundation on collagen mimetic peptides for treating choroideremia. So collagen mimetic peptides, or CMPs, are a synthetic material that repairs damaged collagen and kind of returns it to a normal state. In doing so, it potentially reduces inflammation and oxidative stress. So we wanted to look at these because one of the things that's affected in choroideremia is a basement membrane known as Bruch's membrane, and this thins, and this is where the RPE cells attach. And it's also important for transport of material between the retina and the choroid, and the RPE and the choroid. So we were thinking that maybe the CMPs may help repair Bruch's membrane, preventing potentially the expansion of atrophy, but also act as an anti-inflammatory. Many of you know, inflammation is believed to be an important part of the CHM pathogenesis. We also thought they might affect glial activation, although I'm not talking too much about glial cells today. That is the main focus of my lab. These cells are activated and undergo what's known as remodeling, believed to happen potentially early in choroideremia, and this can affect the way the retina functions and contribute to photoreceptor loss. While some of these changes may be secondary to the initial RPE loss, they're also really important because we won't restore vision if you don't have a normal homeostasis or normal state of the retina. So to undergo this study, we looked at a model that we've developed in our lab. This was actually developed by my late mentor, Jerry Luddy, with funds from Choroideremia Foundation. So in this model, we take sodium iodate, which is a chemical that's known to be toxic to RPE cells. We inject this subretinally, so in the back of the eye, behind the retina, between the retina and the RPE, and we make, as Ben mentioned, a bleb that creates a very small area of atrophy, where we have cell loss. So the top here, these are fundus images and fluorescein angiography. Probably many of you have had this done in an ophthalmology office. So we can do a lot of the same things that ophthalmologists do with animal models. So what you can see, this is a normal on the top, and on the bottom is a rat that we've injected, and we have a very focal area of atrophy. This is just another way to show this. We can look at OCTs as well. You can see on the left is a normal area of the same eye, and on the right is an area where we have atrophy and we've lost a lot of the retinal layers and the RPE cells. So we can look at this, again, look at the animal models the same as what you can see in a human, and it creates a very focal area of loss. And this area actually expands over time. So this is at 3 weeks after the injection, and this is at 3 months, the double arrow showing the area of atrophy. So we see this expansion. So our hope was with the CMP that we could reduce this expansion. So what we did was we did these subretinal sodium iodate injections, and at the same time gave an intravitreal injection of CMP. We then examined the rat eyes at 5 days, 12 days, and 30 days. I'm not going to go through all the data, but just kind of give you a summary. We looked at changes in progression and also in the immune response by looking primarily at the glial cells, and some of this research is still ongoing. So first we looked at the expansion and the thickness. So these are just what we call histology images, and hopefully you can see this is an area here where we have the atrophy in the control, And there's a similar area of atrophy in the CMP. So unfortunately, the expansion or the thickness did not seem to be changed by the CMP, so it didn't affect that progression at all. We then looked at the immune response by looking at the Mueller cell activation. So the cells we look at, the glial cells that we're focused on, are known as Mueller cells. So in this slide, what we've done is we've stained. These are the retina taken in. So we've taken a retina, frozen it down, and then cut it in sections so we can see all the different layers. So normally the cells that we're interested in do not express this protein. So the green staining that you see here is in a different cell. But what happens when these Mueller cells become activated, they start to express this protein, which is known as GFAP. And so this means they become activated. When they do this, they start to lose some of their normal functions, and they're very important for maintaining a normal environment within the retina. But they also release things that can contribute to inflammation and potentially they're possibly helpful at some point, but then can eventually contribute to some of the disease within the retina, and they start to lose their normal shape as well. In doing so, they could be contributing to some of the changes that we see. So we didn't see any change in this glial activation. And we also see a membrane. You can see this line here. The Mueller cells are going to a different shape, a different structure. They actually go below the retina. So we didn't see much changes in those things. But what we did notice when we dissected the eyes was that there was a reduced adhesion. So one thing that happens which we've noticed in looking at eyes with choroideremia and donor eyes that we've received, and also in this model, what happens is the retina becomes very attached to the RPE and the choroid, which makes it very difficult, even in the lab, to dissect the two. And this is a problem when you go to do things like subretinal injections, because it's harder to create that bleb because there's this adhesion between the two layers. But what we notice with these, and this also happens in our model, what we noticed with these eyes and with the CMP, that it was much easier to dissect the eyes, the retinas. So we thought maybe there's a change in this glial membrane, because we think the glial membrane is what's creating that adhesion. And also that maybe there was reduced adhesion protein expression. So I'm not going to really talk about the protein expression, that's still ongoing. But we did look at this membrane. So as I mentioned, the Mueller cells make this membrane below the retina. So what happens in both choroideremia and in this model is, so the Mueller cells normally run the entire length of the retina, but they usually stop before they get to the RPE. What happens when the photoreceptors die, and this happens in retinitis pigmentosa, macular degeneration, and Stargardt's as well, the Mueller cells will come down and create what has been termed a glial seal, and they'll make this large membrane and actually go into the choroid where they don't normally go. So they're going where they don't normally go, creating this strong adhesion. But we see the same, so this is what we see here. So normally this area here would not be green at all. So this whole staining is abnormal, but we see the same membrane forming with the CMP, so that's not reduced. So we're still kind of trying to figure out why we see this reduced adhesion when we still have this membrane. So to conclude this part, so we're still looking at the inflammatory response. Unfortunately, we didn't see a difference in expansion. So this is probably not likely to be a good therapy for CHM. However, the reduced adhesion suggests that it may be beneficial for treating some aspects and maybe making the retina more accessible and the subretinal space more accessible. And it could potentially improve the ability to transplant cells or add stem cell other therapies. And ultimately, it may make a more acceptable environment, because I think ultimately, and not necessarily with CMP, but I think ultimately the ideal therapy that we're going to have for choroideremia is probably going to be a combination of different therapies. So I'm going to switch gears a little bit and talk to you about what we can see when we look at human eyes. So we've been fortunate enough in my lab, and when I was with Jerry as well, we received 2 choroideremia donor eyes. So these are eyes from people that had passed away. And personally, I think this is one of the best ways that we can understand the pathology. We can do a lot with animal models. We can do a lot with cell culture models, but to really understand what's happening in the eye, there's nothing better than looking at human donor tissue. So as scientists, we really view this as a privilege. I mean, when we get an eye in the lab, we really try to we understand this is a huge sacrifice for donors and their families to entrust us, really, and we see it as entrusting us with this precious tissue. So we really try to get as much information as we possibly can out of these eyes. So when we get eyes into the lab, we get them in and we remove the cornea and the lens, and then we can take pictures with a microscope. So these two images are from two donor eyes from people that had choroideremia. And so this allows us to see the pathology that are in the eyes. We then take one eye and we freeze it or do what's called cryopreserve the eye. And this allows us to look at all the different layers of the retina. So this image just shows you what we can see in a normal eye. So there we can really go and stain individual cells. I'm not going to get into the cells, but we can look at individual cell types, different proteins that are expressed. And this really gives us an idea of what's happening, how cells might be interacting, how proteins might be changing, which again can help us understand that pathophysiology and what's really driving choroideremia to progress. We can also take small pieces of this and use it for what's known as transmission electron microscopy. And in this, we can look at ultrastructures and really see very small changes in the tissue, in individual cells. We then take the other eye and we separate the retina from the choroid, and we look at it as what we term a whole mount, where we can actually take it and look at the entire piece. So we can put it on the microscope and put a big piece of the retina or the choroid on the microscope, and we put it on a special microscope that allows us to look at it across it, but then also go and look through individual layers of the choroid or the retina to really understand what's happening in the eye. So this is just on the top is a choroid from a normal patient, a normal donor, and on the bottom is a choroid from a patient with choroideremia. So you can see that there's a large loss of the vessels here, which is expected. This was a very late stage, but this really lets us see this, and then we can take this tissue and then actually embed it into different tissue to look at it in different perspectives as well. We can also look at individual cells. So this I was mentioning that we can look through the different layers. So these two images are actually from the same exact area of the eye. So with our microscope, we can look at one area, which is this, we call it a focal point. And then if we go down into the tissue further, we would get to this area. So we can really appreciate what's happening at a cellular level and just understand more about the pathology. Sometimes we can take the tissue and use it to grow cells, again, to look at how these cells are influencing each other and look at those in a dish. So these donor eyes really help us to understand the pathology and can reveal cellular changes that clinicians can't see. In ideal cases, we can actually look at what the images that clinicians have and match these with what we can see in the lab. And these can help us by looking at the human eyes. We can learn to develop better models and identify specific cell types that may play a role in the disease that we hadn't thought about before. And we can identify different changes and possibly identify new therapeutic targets. So I also just want to mention that it's really important that we understand the female carriers as well. The donor eyes from affected males are extremely interesting and they are very beneficial, but thankfully they're usually very late stage in the disease. So we're getting the end, whereas we really want to understand what's happening in the middle. So female carriers can give us insights into different stages of the disease, helping to provide greater insights. And again, I just want to state that, we consider this a gift when we get these eyes. And it's really a way that anyone can help the future patients and really leave a lasting legacy. And we are really eternally grateful to the donors donors who have received eyes from and continue for the advances that this allows us to make. So I just want to thank the members of my lab and also Stewart Therapeutics, who supplied the CMP, obviously Choroideremia Foundation, and the MDVR for providing the funds for this study, and the eye donors. Thank you.
you.